Microphone is automatically
Is yes, so
linked
it
to.
will pick. Let me see. That one is another.
Yep. That's it. Can you fix it to the?
No.
Yeah. Okay.
Therapy.
Advanced therapy.
Thank you very much for being here. Well done for being on time, and welcome to Tiff and Girls for joining us also. Thank you for signing up. And apologies for the miscommunication about May Hall. We have Professor Yanez here today, lecturing on gene therapy. He is a professor of advanced therapy from Royal Holloway, and we are all very excited to hear what you have to share with us. Thank you very much.
Thank you.
Oh,
I'm very glad. I am very glad that it wasn't me whose mobile phone went off. And thank you for your patience with the setup. I must say that it was actually really good that it wasn't me doing the setting up, rather seeing my colleague, Professor Borger, stressing out here. And I hope you will enjoy the lecture. We obviously have a virtual audience as well as you here today, so I will be trying to accommodate the fact. And thank you very much, obviously, for being here this afternoon listening to me. You will know that this lecture is part of a series given by various academics from Royal Holloway, our institution, on various topics of relevance to the A-level syllabus in biology. And my expertise is in this area of genetic modification of human cells for gene therapy. So I'm going to try to give you an overview of what that actually means, what can be done in this day and age, and why this is a really exciting field of research that you should consider as a career option. So this is Royal Holloway. If you haven't been there, it's a beautiful college in the outskirts of London, not very far from here, and it's worth a tourist visit, if nothing else, if you can spare the time. So this is what I intend to cover in my lecture today as an outline. What is gene therapy? What is a rare disease? Why are those diseases important? The relationship between genes and rare diseases. We will talk about genetic mutations and the new medicine that is coming along and the marketed products in this area. And then I take you a little deeper into the science in terms of what is it that you can actually do with gene therapy. And I will introduce you to the fact that the availability of a medicine in the market doesn't actually mean that everyone can have it, because there is a very big P word in there for pricing. And you will see what those prices are like. And then I will introduce you to an event that we have at Royal Holloway every year. In fact, it's an international event celebrated on the last day of February every year, Rare Disease Day, because every 4 years on a leap year, the last day of February is a rare day. And for that reason, Rare Disease Day is celebrated internationally on the last day of February every year. We have an event at Holloway. We have done it for 12 years, I think, already. And I'll introduce you to what we do for that event over there. Then I will sort of close the lecture showing you some people who are affected by rare diseases, but who are an absolute inspiration, not because they have a rare disease, but because of what they have achieved, despite the fact that they have a rare disease. And finally, I'll give you a couple of ideas for thinking in your own time if you are interested in doing that. Obviously, that is a lot to cover, and there are various styles of painting. Maybe some of you can recognize who the artists who painted these fairly famous drawings are, but I'm not going to do a Dali on the left with all the detail and fantastic figures that he's able to come up with, but it's going to be more like a mirror type painting on the right-hand side. I'm just going to give you wide brush strokes, give you an idea what the field is like, and we will provide also some resources for you to look at in your own time if you're interested, as well as plenty of hyperlinks on the lecture itself if you want to. And my understanding is that these lectures are available online for people to browse in their own time if they want to go back to them later. I should have asked up front, can you hear me well from the back? Yeah, cool, thank you. I can see the tactic that you have used here is very old school. Sit as far away from the speaker as possible, just in case that they ask any questions. And strategically, if you can actually hide behind someone, do it as well. I think you are applying a really old school technique that I can recognize from my time in school a few years back. Anyway, what is gene therapy? We can have loads of different definitions for this. A simple one that I like to use is gene therapy is the deliberate alteration of the genome or the function of the genome in order to produce a therapeutic benefit. So obviously, in order to achieve this, we have to have a fairly thorough understanding of the genome and how that genome works. And I will take you a little bit through that. But in this context, why do I keep talking about rare diseases? Why are those diseases important if they are rare? Why, everyone knows about cancer and cardiovascular disease, diseases that affect most people and are very important for society. Why is it important to study and understand rare disease? Well, there's actually a definition for a rare disease. It's one that affects fewer than 1 in 2,000 people in Europe. 1 in 2,000 people may sound like not very many, but actually there are almost 10,000 rare diseases. We don't actually have an absolute number. New diseases are being discovered all the time, and we add to that number. Take them all together, and they affect 6 or 7% of people. And because there is very little you can do for those people, they actually take a disproportionate part of the health budget, maybe about 20%. In a back of the envelope calculation, 6 to 7% of people, can someone estimate how many that is in the UK? 6 to 7% of people in the UK. How many is that? Go on. But talk to me, not to your colleagues. Roughly, how many people? Maybe on the chat as well. Let's see if you can monitor that. How many people is that, roughly? Is it millions of people? Yeah, it's 3.5 million people. Take it all over the world, it's 300 million people. So these are serious numbers. The way I bring it home is 6 or 7% of people means that if you live in an average road, a few people are going to be affected by a rare disease. In an institution like this, I don't know, maybe 1,000 students in the ballpark, 60 to 70. In our college, in a community of 15,000 people, hundreds of people affected by rare diseases. You may not know because many have hidden disabilities, but they are there and they are important. In addition, many of these rare diseases affect children, and 30% of those, as an estimate, will die before age 5. So they are very severe diseases. And the majority of them are inherited. They are passed on from parents to children. In addition, because we are growing an understanding of those rare diseases, we know what gene is faulty. We are starting to understand what the effect of that fault is. We, in principle, know what we have to do to treat the disease. In principle, we may not be able to do it in practice, but in principle, we think we know what we have to do. In technical words, the target for the disease has been defined and potentially validated. And even more important, the therapy technology that is being developed in the field of rare disease is now being applied to common diseases once we know what we have to do. And I will touch on one of those cases when we are now able to treat leukemias very effectively after those people affected have gone through the standard treatments. And effectively, there is nothing else that the standard medicine can do for them. Now we have, specifically for some leukemias, some fantastic treatments that actually can put those people in remission. And that is using technology that was originally developed in the context of rare diseases. So this is globally why those diseases are so important. At the individual level, because these diseases are rare and unknown even for professionals in the medical field, diagnosis of these diseases can take a long time. In average, about 5 years. Imagine having a child who is affected by a disease, or a sibling or a relative who over 5 years sees an endless succession of specialists who are unable to diagnose the problem with the child. As I say, in the field, on average, 5 years to achieve a diagnosis. And then very complex care, which, as described on this report by an organization called Rare Disease UK, should be done in centers of excellence that have all these categories. But actually, this is not the way that is currently done. This is a goal to be able to provide care which is coordinated and long-term as described here, but it's not how it's done currently. Why do we talk about these things at Royal Holloway? Well, we have an interest in developing this technology, and we work in several rare diseases, spinal muscular atrophy, ataxia-telangiectasia, which probably you haven't heard of before, muscular dystrophies and atrophy. I guess that all of you have heard of motor neuron disease, and there have been some very high-profile cases in the news, including a lot of funding from government for a specific motor neuron disease center on the back of a high-profile campaign led by Rob Barrow, who will be familiar to many of you as a rugby player who developed motor neuron disease. But as I say, this technology is advancing, and we are hoping to apply it to common diseases as well, or more common diseases like spinal injuries, Parkinson's disease, perhaps Alzheimer's, and you can treat bacteria and viruses with this. And I will touch on the fact that some of the main vaccines that we have for COVID-19 have been developed using gene therapy technology. So I'm going to take you back a little bit and go into the basics. I know that you are very clever boys and girls, and you, I'm sure, know this, but just in case, let me give you a refresher of what those genes and proteins are. So you know that we have a set of chromosomes in each of our human cells, and those chromosomes contain many, many different genes, roughly 20,000 genes. And a gene would normally produce a protein, and that protein will have a particular activity or several. Some of them are enzymes, some of them are structural proteins, and so on. But then in some cases, you can have a mutant gene. And we'll discuss a little bit this concept of mutation. And if you have a gene with a mutation, which in this slide is represented as a green blob, perhaps you have produced a protein that is either abnormal or maybe not be there altogether. And that may or may not have an effect. Here highlighted this at the particular location of one of those genes in the genome on the 2 chromosomes from mom and dad. And there are 2 copies of the gene highlighted in green in each chromosome because this is after the S phase, the synthesis phase of DNA. So each chromosome has 2 chromatids. So you get this dot highlighting the location of the gene in that particular case. And it's only, I'm pointing to one location out of 20,000 in the genome, which may or may not be a problem. So You, I'm sure, have heard of mutations. Mutation is a word that is becoming not politically correct. And we tend to talk more about variation because we are all different. In reality, we are all mutants, right? There are no normal people and mutant people. We are all mutant. We are all different. We share, say, general sequence for the human genome, but each of us has a specific differences. So if the genome of each of us is compared, about 1 in 1,000 bases in that genome will be different between any two of us. That's a lot of variation. So I'm telling you about mutations that can lead to genetic diseases. Are all those mutations bad? Well, some are irrelevant. The fact that you have a mutation or a change in a particular gene is irrelevant. Maybe because in that particular position is not important, maybe because there is another protein which makes up for the function of the mutated protein. And in some cases, it's minor. Those of you who are closer to me may notice that my two front teeth are quite separate from each other. And that is because genetically, I'm actually missing two teeth. So there is more space for the rest, and they have spread out. So that's, you know, it doesn't cause me a problem other than when I was a teenager like you. Obviously, I would have liked to be tall and blonde and have perfect teeth. But in actual fact, is not an issue, right? And evolution that allows nature to adapt to a changing environment is based on mutations. In other words, not all mutations are necessarily bad, but some of them can be. And a single change in the DNA can lead to a very severe disease. And I'm just highlighting two of the diseases we work with at Royal Holloway: Duchenne muscular dystrophy and spinal muscular atrophy. Both of them are extremely severe. Duchenne, in most cases, leads to death in the late teen years, or maybe 20 odd, from breathing problems. And a spinal muscular atrophy, in the most severe cases of type 1 spinal muscular atrophy, children die by age 2 at the latest. So it's a little bit of genetic lottery. A single change in your DNA, it can be from irrelevant to a massive phenotypic effect and a very severe disease. And just to give you an idea of the task of, I've told you that there are almost 10,000 rare diseases. We don't actually have a number. There is a formal list, and you can see the names of those diseases. I'm not even going to try to pronounce them. But to give an idea of the task, do you know if you were going to read aloud the list of rare diseases, how long would it take you to read it? Any idea? Give me some time and see how close you are. I heard a couple of hours. It will take you longer than that. Talk to me, guys. Talk to me. How many hours? How many hours? Or how many days?
7 hours.
More. Less. 2 weeks. Less. I'll shorten your pain a little bit. If you did it non-stop, 18 hours. So just literally reading the list of rare diseases non-stop will take you 18 hours. I can tell you that people can spend a lifetime trying to understand each of those diseases in order to try to perhaps develop a therapy in time. So it's just a monumental task ahead of us. But luckily, there are some trailblazers, and I'll show you some examples in which there are treatments for them. But the task is monumental, okay? And this is a little bit highlighted by the fact that there's a very big gap between the number of rare diseases, I told you perhaps close to 10,000, and the number of diseases that a child born in this country is screened for at birth. And perhaps some of you know how many diseases that is. So every child born in the UK will have a drop of blood taken, and that blood will be screened for a number of genetic diseases. Do you know how many that is on average? Not far, but more than five. It's actually officially nine, but depending on the health authority, there is some variation. In some cases, 20 odd. But there is a massive difference between the ones we screen for and the total number. The reason for that is, in many cases, knowing early that a child is affected may not make any difference in practice because there is very little to offer them. But that is changing quickly as we have more and more treatments available. So this may be something that is going to change. Also, because the technology is developing very quickly. And perhaps you have heard of the 100,000 Genome Project and a government quango called Genomics England. Has someone heard of Genomics England? No one? You need to start reading the papers. Okay? England is at the forefront of genomic sequencing and genomic analysis in the context of disease. And a lot of the human genome sequencing was actually done in the UK at the time, particularly at a center called the Sanger Center near Cambridge in Hingston Hall. And the government is very keen on this technology, and I will show you on the back of that some official documents that have come up. But the technology has developed. It is possible for a relatively modest amount of money to have your full sequence genome determined. You can actually do it privately. There are companies that for roughly £1,000 will sequence your genome and will give you the sequence and some information about disease risk. So the technology is there. Those places where the sequencing is done look very much like factories, many more machines than people, because determining the sequence is not so complicated as actually analyzing the sequence. The holdup at the moment is on the analysis, but it is possible. So in principle, to advance in terms of diagnostics is possible. And some of those mega projects, particularly in the UK, are trying to use genomics to diagnose people who are affected by some of these rare diseases, and cancer particularly are the fields of application. So this is a critical area for the UK. The government sees it as that, and two documents have been produced. One of them is Genome UK, the future of healthcare, and the other one is a UK rare diseases framework in which the government has outlined their vision to promote this type of research in the UK for the application of the technology, for the development of the technology, and ultimately for the benefit of people affected by these diseases. Those hyperlinks to those documents are available on the additional resources that are provided with the masterclass, and you can go to them in your own time. I just want to highlight that this is important nationally. This is part of the business strategy of the government to promote these areas of research. Oops. And genomics is now an integral part of the NHS, is now being used for diagnostics. When children are born and affected by a disease that is not immediately diagnosable, in some cases, not in every case, but in some cases, genomics technology is applied to try to help them. There are scientific papers demonstrated that, again, for people who are in intensive care, in some cases, genomics diagnostics can help to pinpoint what the problem is much faster than traditional diagnostic methods. So again, there is a drive in this country to embed genomic services in the NHS. And this slide was initially produced in 2018. So we are now 4 years later in the advancement of this. As you can imagine, big changes are happening in the NHS to accommodate this. And I told you that for some diseases, there are marketed treatments based on gene therapy. And those treatments come in 3 flavors. In some cases, the treatment is what we call an antisense oligonucleotide. Perhaps the word oligonucleotide rings a bell. Who has heard that word before? Oligo or oligonucleotide. Have you heard it before? Yeah? Okay. They are short sequences of a nucleic acid that may have a therapeutic effect. So some of the therapies for the diseases that are listed there, Duchenne muscular dystrophy, spinal muscular atrophy, and hereditary ATTR amyloidosis, are antisense oligonucleotides, which are able to alter the function of the gene to achieve a therapeutic effect. The second flavor of these treatments is viral vectors, so engineered viruses that are able to deliver the gene that is faulty in people affected by the disease, or viruses that perhaps can kill cancer cells, what we call oncolytic viruses. And the third flavor is genetically modified cells. You perhaps may be surprised, but in some cases, what is approved for marketing by the regulatory authorities is actually a genetically modified cell, which has been modified with a gene that may have a therapeutic effect. And in some cases, that is for cancer, like in the cases of Kymriah and Yescarta, where you have T cells that have been modified to express a gene that can kill leukemic cells, or in some cases, the genes express the faulty gene in the person affected, and in that way, rescue the function. So this is online. These are marketed treatments in the US and in the EU, even though there is some coming and going, so to speak, because the fact that a treatment is marketed doesn't mean that it stays on the market forever. And the 2 that are in brackets here made it all the way to marketing, but for commercial reasons, the company eventually pulled them out of the market. And I will give you an idea of why that may be the case. This is no longer a complete slide because more and more are being approved. So if you are interested in your own time, you can go to that hyperlink, which will take to a site maintained by the International Society for Stem Cell Research, and they keep tabs of all the gene and cell therapies marketed across the world. I told you that I would give you a little more detail of what you can actually do with gene therapy. And that is a little list of what you can do. You can deliver what we call a mini gene that will make up for the loss of function of the faulty gene in the person affected. Or we can alter the function of a gene to make it produce more or less protein. We can kill cells, and you don't have to be very imaginative. Why would we want to kill cells in a person? In what disease type? Cancer, right? So that's the one example in which we may aim to kill cells. We may want to actually go for gold, repair a faulty gene, as opposed to replace the function with a different copy. Surprisingly, we may want to actually stop a gene from working altogether because that may lead to a therapeutic effect. And as I told you already, we may want to vaccinate, as in the context of COVID-19. So I'll take you through those avenues with not much depth, but at least giving you an idea. And most of these strategies require the delivery of genetic material to cells. But cells are very well designed to prevent the delivery of genetic material, because normally incoming genetic material would be a virus trying to infect the cell, and cells don't like that. So they protect themselves very well to prevent the entry of nucleic acids. So we have to figure out ways in which we can overcome that. And very grossly, those are classed in non-viral or viral methods for gene delivery. My lab uses a lot of viral methods for gene delivery, because viruses have evolved over millennia to enter into cells efficiently and deliver the nucleic material to those cells. So we effectively hijack the viral machineries to deliver our transgenes, the genes that we want to deliver to the cells of interest. And very generally, there are two approaches to do this. You can either treat the person directly, what we call an in vivo treatment, or we can do an ex vivo approach. And that ex vivo approach means that we take cells from the patient, we genetically modify them in the lab, we quality control them to ensure that everything has gone according to plan. And if that's the case, we transplant them back into the patient. And when I say we, I don't mean my research lab, I mean the clinical community delivering this as a therapy in hospitals. And that's what we call an ex vivo therapy. All those genetically modified marketed treatments that I showed you are examples of this ex vivo strategy for gene therapy. So how do we go and do these different strategies? So how do we introduce a minigene? Generally, we use an engineered viral vector to do that. We do that, as I say, because viruses have evolved to very effectively deliver a nucleic acid cargo onto cells. So we engineer those viruses, and we call them viral vectors once they have been engineered. And we have replaced the genetic material from the virus with the transgene that we want to deliver to the cells of interest. The first disease in which this was considered an absolute success was a severe combined immunodeficiency, or a SCID, which I'm sure is a term that most of you will have heard in some context, but perhaps you are more familiar with the boy in a bubble disease name. As these people do not have a working immune system, they are very prone to very severe infections from multiple organisms. So traditionally, they lived in a protected atmosphere. And in the context of this particular disease, people were able to genetically modify a virus with the gene that is faulty in people affected, deliver that virus to the relevant cells, what we call hematopoietic stem cells, so the stem cells from the blood in the lab, check that the genetic modification had gone according to plan, and transplant those cells back into the patient. And in most cases, if you see the list of outcomes at the bottom, that was successful. And that means that those children live, while in most cases, most of them would have died. In some cases, because subjecting them to a bone marrow transplant from a mismatched donors can have some or very significant side effects or adverse events leading to death. So this was the first case in which a gene therapy was unequivocally demonstrated as a success. It's not that it came without hiccups, but is the traditional example of gene addition therapy. Second strategy, to make a gene produce more or less protein. Why would that be therapeutic? Well, in this case, the example is one of the diseases that we work with at Royal Holloway, spinal muscular atrophy. In this particular disease, people who are not affected have two very similar genes. One is called SMN1 and the other one is called SMN2. SMN1 makes a lot of normal protein, SMN2 makes very little. People who are affected by spinal muscular atrophy have a mutated SMN1. That gene doesn't work for them. All they are left with is the lazy copy of SMN2, which makes very little protein. And they struggle with that. They develop spinal muscular atrophy. So scientists have figured out a way to increase the level of normal protein that can be produced from this lazy SMN2 gene. And for that, we use one of those short oligonucleotide sequences that targets the RNA produced or the messenger RNA produced from the SMN2 gene and creates or targets it for a modification that boosts the levels of protein produced from it. So this is a therapeutic approach for that. And it may sound completely hazy. You may need to look that on paper to understand the process. What difference can that possibly make to a child? Well, this is a real child called Cameron who is affected by spinal muscular atrophy, type 1. Normally, he would have been dead by age 2 at the latest. But he was one of the first people treated in a clinical trial with this oligonucleotide called Spinraza that can change the processing of the messenger RNA of the SMN2 gene. And Cameron was able to achieve developmental milestones like sitting up unaided and standing up that were unheard of in people affected by type 1 spinal muscular atrophy. So this type of molecular technology can have a real impact on the effect on the disease on people. Cameron is not fully cured, but certainly is gone much, much further than other children of that age affected by the disease, who, as I say, in the most severe cases, leads to death by age 2. Killing cells as a therapeutic strategy. I've introduced to you that this is chiefly important in the cancer field. And I've already mentioned that in the context of leukemia, we are applying gene therapy technology that was developed for rare diseases and now is being applied to a common disease like cancer. Leukemia, at the end of the day, is a type of liquid cancer. And in this case, what has been done is take T cells from the person affected by that leukemia and introduce in those T cells a gene that is able to target the cancer cells for destruction. And the way that is done is with a modified antibody molecule, here highlighted in green, that will bind a protein expressed by the cancer cells. In fact, by all B cells. That protein is called CD19. So these T cells are now targeted to CD19 containing cells, and they will kill them without any control from the histocompatibility complex and the need for antigen presenting cells. And for people affected by certain types of leukemia who had been through all the standard therapeutic avenues, this has been a lifesaver. Repairing a gene, as I say, the gold target in the field. This was a goal for many years, and the technology was available, but it was actually very inefficient. So very few cells in a population of cells would undergo the correction modification that we were aiming for. And relatively recently, something called CRISPR-Cas came along. Who's heard of CRISPR here? There you go. You have heard of this technology, which is in principle something obscure coming from very rare studies in bacteria. And this is because CRISPR has made it so much easier to introduce those defined genetic modifications in the genome, including the possibility of repairing genes. So CRISPR is different to other technologies in the sense that is so easy that most labs in the world doing molecular biology are using CRISPR in one way or other. We can just buy the reagents very easily, and within days, you can be applying CRISPR in your lab. While doing this type of what we call genome editing, traditionally was much more cumbersome. You may take years, literally, to develop the technology to change one particular location in the genome. So with CRISPR, what we can do now much more effectively is introduce defined modifications. Very generally, we deliver a gene with the correct sequence together with the CRISPR molecule that can cut the target genome at the place where we want to introduce the genetic change. And the fact that the chromosome has been broken at that location boosts the frequency of modification massively and takes it to therapeutic percentages. So this, in principle, is now in clinical trials for treatment of disease. Stopping a gene from working as a therapeutic avenue. Again, that is not immediately obvious. Why would that be the case? And how do you do that? Well, you again use CRISPR technology to do that. You can, if you introduce a cut in the genome, the cells will try to repair that because breaking a chromosome is quite a problem for the cell. So cells have mechanisms to repair that type of damage. And in repairing the damage, they often introduce very small changes in the original sequence. And those changes can lead to the disruption of the gene, which no longer works normally. The first therapeutic example in which this was demonstrated as a therapeutic avenue was in the context of HIV AIDS. You may know that for HIV to enter the T cells that it infects, it binds a particular surface molecule called CD4 and other molecules on the surface of the cell called the core receptors. And people demonstrated that altering the sequence of the core receptor for HIV would greatly minimize the infection of the cells with HIV. So the destruction of the gene for a HIV core receptor has been explored as a therapeutic avenue in AIDS. And finally, and you all have heard of this, even you perhaps haven't realized, a lot of the vaccines, obviously, we have a lot of different vaccines for coronavirus, luckily, because no one could have predicted upfront whether we would be able to have a vaccine for it. You know, we know about HIV AIDS for 40 years, we still don't have a vaccine. We are quite blessed that within a year of COVID-19 developing, we had vaccines for it. Those vaccines have been developed using a variety of methods. You can see there, some of them are DNA or RNA, some of them are live attenuated vaccines. So the virus has been disabled in some way, but it's still a live virus. In some cases, it can be an inactivated one completely. In some cases, can be just parts of the virus, what we call subunits. And in some cases, it's a viral vector. So some of the vaccines that have been most used in this country, in fact, the ones that have been, the Pfizer and Moderna vaccines are of this type. They are RNA type vaccines. And the AstraZeneca is an engineered viral vector, a type of vector that normally would cause a cold. So, you know, most of us have been transiently genetically modified for us to become immune to COVID-19 using technology that was originally developed for genetic therapy. I told you that the availability of treatments doesn't actually mean that people have access, particularly across the world. And that is because these are very pricey treatments. Spinraza, that oligo that can correct the spinal muscular atrophy, has to be administered repeatedly to people affected, 6 times in the first year, 3 times every year thereafter. You can see the cost of a dose, that's the cost of the vial, and the cost per year in the first year and thereafter. One particular genetically modified cell treatment costs effectively £500,000 per patient. The most expensive medicine ever so far is a viral vector, so an engineered virus to treat spinal muscular atrophy, expected to be a one-off treatment, but at a cost of at least list price, catalog price, essentially £2 million. As you can imagine, you hear that our NHS is stretched to the limit, providing a standard treatment. If we have more and more treatments for rare diseases at these prices, this is not sustainable. And the industry and governments are looking for new ways to fund these treatments in a way that they can be applied to people. At the moment, the treatments are only available, as you can imagine, in developed countries, and within those, in particularly wealthy countries, to some people in the population. That's not fair. So a very important goal for all of us is to ensure that these treatments become available for all indications and across the world. And that is quite a challenge. Naively, one would think there is a treatment available, problem is sorted. Actually, as you can see, that is not the case. And pricing in this particular case is a very important issue. In the context of the development of the technology and those prices, you can imagine that the general market value of this technology is on the increase. And that's why there is so much interest, as you can imagine, from governments around the world to become leaders in this area of regenerative medicine, if you want to call it that. And wrapping up, I told you that there is a Rare Disease Day celebrated on the last day of February every year. At Holloway, we have celebrated it for 11 years. This February will be our 12th. And we do a variety of things. We normally actually host students at Royal Holloway. So people like you from secondary schools doing A-levels or advanced GCSEs will come. They will listen to presentations. They will visit an exhibition. They'll do hands-on activities in our labs. And they will chat to our undergraduate students to discuss any common topics of interest, and do various also activities in the labs. For the last 2 years, that has not been possible. Last year, we had to move to an online system, but we tried to do very much the same. We obviously had presentations, that's the easy part, but we also organized remote activity with the students doing that wherever they happen to be. And we also organize those chats with our undergraduates at the same time. And for this year, we are working towards the same. So we will have lectures, we will have a remote exhibition, we will have interaction, what we call a speed dating session with our undergraduates. And in the afternoon, we are providing the information for schools to set up the activity if they are able to join us. So that may be something that this school and all the other schools that are listening remotely may want to consider. All the information is on the hyperlink provided on the slide. And finally, I told you that people affected by rare diseases can be really inspirational. Not because they are affected by a rare disease, they will find that infuriating, but because of what they can achieve despite the fact that they are affected by it. We had a patron for our 2012 event. 2012, you will remember, was the Olympics in London. Our patron was a student from Royal Holloway, who is a Paralympic gold medalist in rowing, Helene Rageford. And at some of our events, we have had people like Srin Madipali, who is affected by spinal muscular atrophy, but that hasn't stopped Srin from going pretty much everywhere that you can think of. And in fact, he's the founder of the disabled Airbnb, which was purchased by Airbnb, and Srin, a British person, moved to California to enjoy the life there and promote his variant of Airbnb in the context of Silicon Valley, basically. So, almost my last slide. I told you that this is going on. Genetic therapies are no longer a chimera. We have some, and there are many more under development. So this is a really exciting area for people to consider as a career move. This is in many different aspects. If you want to be a clinician, by all means, that may be one way. There is plenty of research being done in labs to develop the technologies. And if your interest perhaps is in computer science, as I told you, there are plenty of data to be analyzed coming from the study of the data of all those genomics technologies that are coming online. So there are loads of areas in which you can contribute to this genomic medicine revolution if you want to consider that as a career. These are two questions that I'm throwing out there. If you want to do some thinking in your own time, you may want to consider how you would choose a virus to make a new gene therapy vector, or whether you would sequence the genome of every newborn to diagnose a genetic disease. I'm not asking for answers now. This is for you to think in your own time. And I understand that in the context of this masterclass, often the week after, there is an opportunity for discussion with the speaker. So you may want to do that. And finally, I know that there are many things that you could be doing rather than listening to me here. So thank you, but you are all underage, so forget about those two. And thank you very much for your time.
Thank you so much, Rafael. So now the exciting part is the questions. Now you need to train your brain and come up with some questions, please. Yeah.
You mentioned about how you use viral vectors to do the whole genome thing. How do you? Sorry, that was really big. But how do you engineer a virus? Like, how do you change the structure of the virus?
Yeah. So the question is, how do we actually engineer a virus in practice to convert it into a viral vector? We need to know about the biology of the virus. We need to know the disease that it may cause. Some viruses cause disease, like HIV, like the viruses that cause the common cold. Other viruses infect us but do not cause us any disease. So we have to have an understanding of the biology and the pathology of the virus. But chiefly, we need to understand the genetic structure of the virus. So what genes does that virus encode? What is the function of the different genes? Are some genes that are particularly involved in pathogenicity? Are they the ones that cause the symptoms of the disease? So generally, we need to understand all that. We need to remove all viruses that can have a detrimental effect on the person, so all the pathogenic genes. And we have to introduce the gene that we believe is going to be therapeutic. So basically, it's a replacement mechanism in which we understand, we identify anything that can be detrimental, remove it, introduce what we think is going to be therapeutic, and test. And this is literally a life of trial and error until we can develop a technology that is effective in the particular cells of interest. But the basic technology that we use is genetic engineering. We can effectively cut and paste nucleic acids to make the fine alterations, and that allows us to engineer viruses as well. Thank you.
Questions?
Please. Okay. So the question is, what makes gene therapy so expensive? Why are they so special? There are various angles to that. One is they are new, and often they replace conventional treatments that are very expensive. So the equation that companies developing these therapies do is we have a new treatment, which perhaps can be a lot better than a traditional treatment that in itself was very expensive. And we don't actually know for how long we will be able to market this therapy. So the point is, let's try to make as much profit soon as possible. But companies do not actually publicize how they decide on the pricing of the therapies. This is all guesswork. So it's new technology, it's applied to few people, and companies are concerned regarding the potential time frame of application of their novelty, and they charge very high prices upfront. It is true that the technology is developing all the time. The fact that we can apply it to more and more diseases means that the market is increasing. Oops. And that also means, don't worry, let's see, I can pick it up later. That also means that more companies are developing the technology to increase the scale so we can make more of the same viral vectors, more of the same chemically synthesized molecules, and there is economy of scale. So the hope is that eventually those prices will come down seriously. But at the moment, it's being an issue. There are some treatments that in principle could be marketed, but they have not been approved for marketing. So for instance, one of the treatments I listed on that slide of marketed treatments, the one called Livmeldy, has been approved in the EU, but the organism that approves therapies for marketing in the, not for marketing, but for use within the NHS, that you may have heard of, is called NICE, NICE, has not approved the therapy for delivery through the NHS. They consider it too expensive. So even in a highly developed country with, in principle, the resources available, some of those marketed treatments may not be available. So this is a real problem, and it really has to be dealt with. So the hope is with economies of scale, with more companies competing for the technology, perhaps providing treatments for the same disease, then those prices come down. But really is a very big issue because we are having all this in the context of an NHS or national health services in all the developed countries which are at breaking point. And you know, I always say that there is only one thing that can be more sad than having a child affected by one of these rare diseases, which would be having a child affected, knowing that there is a treatment and that they can't have it for whatever reason. Imagine, what those families must think.
I have actually a related question. I was thinking very much on the same way, but just let's phrase it in a different way. So, how much you think this price is the development of the treatment, the research essentially, which is done by academics and the companies and ended up in a patent? And how much portion of this price might be just the simple manufacturing of the reagents and performing the treatment?
So lastly, Professor Bogre is asking how perhaps that pricing is worked out, how much it can be the development. So really figuring out what can be a therapy, doing the lab development of the therapy, all the testing. Afterwards, you have all the clinical trials to test it in people. Then you have to manufacture it large scale. Companies do not make that information available. So strictly speaking, we don't know. I am very critical with the pricing policies because as LASI was advancing, a lot of these therapies have been developed in academic labs, funded by universities, funded by small charities, funded by the government. Once those therapies come to a stage where they need to be tested on people, that is quite expensive. And normally companies take over at that stage. And then they keep all the intellectual property and determine the pricing, the manufacturing, and everything. So the people who have actually developed the therapy at the start lose control and companies take over. And if there is no competition for that particular disease, that's the only treatment. In a way, they can charge whatever they like. You know, again, the way I put it, if I was a maker of chocolate and I said, I make the best chocolate in the world, it's going to cost you £2,000 per tablet. You would send me packing and go and buy a Cadbury's tablet for £1. Okay. But if I have a treatment for your child and it's the only treatment available in the world, and I tell you that's £2 million, that's a different game, isn't it? Imagine the pressure that those families can put on the government, on the NHS, on insurance companies for that treatment to be made available. That's the game that is being played, right? It's an ugly game. I don't like it. And I've been fighting in the context of a number of organizations for that to be changed. But at the moment, companies are in control. It's complicated, but at the same time, companies wouldn't be interested in developing a therapy that they cannot market. So it is in their best interest that those therapies are marketed. I think that marketing will require that price coming down. The availability of competition is also very important. As an example, one of the diseases I mentioned, spinal muscular atrophy, is very unusual in the sense that being a rare disease, there are 3 marketed treatments for it. One is a small molecule, which I haven't mentioned. One is one of those antisense oligonucleotides, and one is a viral vector. Now there is real competition because they are made by different companies. Roche, Biogen, and Novartis are the 3 pharmaceutical companies producing those treatments. But even there is an issue because they don't really want to do head-to-head competitions of the treatments. We know that the 3 treatments separately are effective, but they don't want to do head-to-head just in case their own product comes worse off and then fewer people buy it. So, I mean, there are all sorts of politics going on. But I think that in the long term, for viability of the field and for those therapies to be available, the prices will have to come down. There is no other way, simply. Families can put a lot of pressure. I told you that there are 3.5 million people affected by rare diseases in the UK, 300 million globally. And as more therapies become available, the pressure from those people can change things.
So any further questions now and in another topic? If not, then let me just say a couple of other words that we do want you to be active on this masterclass. Yes, so this is not a cinema type of activity, but we want you also to work. So are you going on the Padlet forum this Yes, so we are putting up because the essence of science is to immerse yourself in the topics of so we are trying to provide you a variety of topics and also trying to give you like the questions Raphael put up and I will also put it up on Padlet and you can work on them in the comment box, but also a lot of other resources. So the other thing is that you need to read. Yes, so to get into the science, you need to read. So are you taking New Scientists? Reading? No? That's good. That's great. Fantastic. At least some. So this is something which you need to pay, but they are not really expensive, couple of pounds a week, right? But I also put up a reading material which is called Science Daily. So this is on the Padlet link, which is an internet-based free resource of good people, editors collecting research all over the world and putting it up there freely. So if you want to get news, what is the most trendy in science, what is the big new discovery in science, then that's Science Daily. Yes, so you can also put up little alerts for yourself on your own topic and things like that. And also, if you are making some courseworks, or not courseworks in your case, but EPQ or any kind of other type of works, then Googling aimlessly in a browser, or trying to use the search in a Science Daily webpage, which is filtered to good papers, is in my opinion much better. Yes, and we are also preparing for our symposium. So you need to find topics and gang together a group of 4 and start working on a topic and the symposium. And the last bit to this Raphael question on the price. So we on the symposium we are giving prizes for the best poster, but now I give a prize for the best question, which was this prize to the what is the cost of this gene therapy. And this is a book by John Salston. So he's a Nobel Prize winner who is extremely passionate about exactly the same topic which we were discussing, that how policy can be changed, that governments buy these patents, for instance, or what kind of other solutions we can get that treatments are not denied to people. And Salston actually is a honorary member of our college. He passed away a couple of years ago. He works on C. elegans and got his Nobel Prize for how cells die, which is important for many things, including cancer. But so this is